preparing, and as soon as as we are live, I will let you know and end the stage to Christine.
Okay, sounds good.
Yeah. Okay, so now we are live. Christine.
Okay, so do we have an audience yet?
Yes, people are joining. At least I have 2 watching now.
Wow.
Maybe we wait a minute, because it would be kind of a shame
we
if
can
people
wait.
would be a shame if most people miss the start of the talk, right?
Yeah, we can wait 5 minutes,
Well,
I guess.
I don't know how quickly people join. I mean, I can't see it at the moment. Okay, I guess we can just start now. So people have arrived, so that's good. So welcome everyone to the closing keynote of EvoStar 2021. It's my great pleasure to introduce our second keynote speaker, Professor Susanna Manrubia from the Spanish National Center of Biotechnology in Madrid. And as some of you might know, Susanna has a background in physics, and she holds degrees from the University of Barcelona and the Polytechnic University of Catalonia, respectively. And after her PhD, she actually spent several years as a postdoc at leading research institutes in Germany. And most remarkably, she held one of the prestigious Humboldt fellowships funded by the Alexander von Humboldt Foundation while she was in Germany. She then moved back to Spain to join the Center for Astrobiology in Madrid. And now she is an associate professor in the systems biology department at the Spanish National Biotechnology Center. And she leads the evolutionary systems research group there. So looking at her CV, Susanna has published over 130 peer-reviewed articles and 3 books, and she has served as editor or section editor for leading journals in the field, for example, the BMC Evolutionary Biology Journal, and she is also a founding member of the recently introduced journal Virus Evolution. Her research is transdisciplinary, so her research focuses in particular on problems at the interface of physics, biology, and society, so nicely combining several areas and in particular her background in physics. And there she investigates the emergence and effects of evolutionary mechanisms, including cultural patterns and collective social behavior. So, and if you had a look at her abstract, then today she will particularly talk about genotype-phenotype maps and evolutionary dynamics, which I believe is a highly relevant topic for our research community. And I really hope that we will get some very interesting novel perspectives in this area. So I would like everybody to join me in welcoming Susanna to EvoStar. And Susanna, the floor is all yours.
Okay,
so
thank
very
you.
much forward to your talk. Thank you very much.
Thank you very much, Christine. It's a great pleasure to be here. For me, it's a new ghost audience. So I didn't know much about your society, but I feel after looking at what the things you do, the things you're interested in, that there are indeed interesting points of touch with the research that we pursue currently. So it's an opportunity for me also to convey our last achievements and the viewpoint that we entertain regarding this area, right? So it would be much better to be in Seville with a beer in the hand and to have time for a chat afterwards, but this is how it is. So I hope that's not too boring, that you just watch me and I watch the screen while talking. And so I'll be happy to answer any question at any time. So if anyone wants to interrupt, it's okay. It makes it a bit more close. This kind of talks, right? So without further delay, I will share my screen. Let's see if this works. Here we are. Can you see it?
Yes, I can see it.
Yes, is this fine? Okay, so this is the title of the presentation. I'm going to talk about how the architecture of, you will see, hyperastronomically large genotype spaces shapes evolutionary dynamics, right? And I'd like to start by talking about this young man. This is Lewis Carroll. Lewis Carroll was a mathematician, but he was also a person well known in his time because he liked it very much. So crosswords and all kinds of entertainments based on words and math. So it was a mixture of both worlds, right? So one of the inventions that he had was this kind of this problem that it was known at the time as doublets. Right, so this is a word puzzle that he himself explains that he invented it on the Christmas day, almost half a century ago, for 2 bored young ladies who had begged him to send them some riddles. So having none at hand, he instead invented this game, which he originally called word links. So as you see, and you may have played that, so the game is that you get 2 words and you are allowed to make, say, point mutation, so to change single letters such that you go from one letter from pig to sty, right? By changing just one letter at a time. And you have these chains that allow you to go from one to the other. And that was the game. You're given the 2 words and then you have to connect these 2 words. Okay, why are these doublets fun? So can we always solve them? So if you look at the dictionary of the American Association of Scrabble Players, you see that there are a total, so let's focus on 4-letter words, right? there is a total of over 4,000 four-letter words with meaning, right? But if you look at the number of different sequences of that length that you can form with an alphabet of 26 letters, then you get almost 500,000 of different sequences. And among these 500,000, so slightly less than 1% has meaning, as we see here, right? So if each letter, 4 letters, has, you can mutate each position in 25 different other letters of the alphabet. So every 4-letter word has 100 neighboring sequences, but only less than one neighbor on average has meaning. So the question is, how is it so that that we can solve these riddles? Because if I have less than one neighbor on the average, this means that 1 out of about 10 times, I will not find the neighbor for a given word if meaningful words would be so put at random, also occupy random positions in this total of the 4-letter words with meaning. Is that clear? So I would be playing, and very often I would find that I have no neighbor with meaning, so the game would stop. So instead, it seems that it can be systematically be solved, and it is fun. It is fun because some of the words are rare, but you eventually find them. So what's the sort of answer to this apparent paradox of an unsolvable game? Well, the point is that meaning is not randomly distributed. In any language that you consider, if you think of 4-letter words, these should be words that you can pronounce, that you can utter, right? So essentially, they will have more vowels on average. They should have at least one vowel generally. And when you make this connection, this is a work of so a few years ago, where people, what they did was to take every 4-letter words and look at how these words distributed in a network. So the number of neighbors they had following this principle of changing one letter and having a meaningful neighbor. Okay, so what you see is that most of the 4-letter words in English form a large connected component, right? So you can go from any place to any other place. Okay, you have to be clever or know enough English to find the pathway, but this pathway exists, right? To go from one to the other. And you have very few instances of isolated words. I don't know, like twip or so weird words that do not have any neighbors. But the vast majority are connected. And if you look at the distribution of the number of neighbors that each word has, you see this kind of distribution, right? Where this is decreasing. So most words have 1, 2, 3, 4, 5 neighbors, and then it decreases to rare words which have over 20 neighbors, right? But this is the essence of the game. Meaning is not randomly distributing, it's correlated. So if you have a 4-letter word with vowels, it's more it's easier to find a close word that is also meaningful, that you have an instance of 4 consonants, for instance, right? So you have these correlations. Why am I talking about this? Well, because this kind of metaphor was later used by John Maynard Smith, which is probably the most brilliant theoretical biologist of the 20th century, when he illustrated how sequence spaces can be navigated. So he was responded in his small piece of the 1970 that was published in Nature, that if evolution by natural selection is to occur, functional proteins must form a continuous network which can be traversed by unique mutational steps without passing through non-functional intermediates. He was answering a paradox, an apparent paradox that was proposed by Salisbury 1 year after that, so in 1968 actually, and he was contending that if you have just random sequences, it's impossible that you never get a functional protein. So he had in mind the idea that one sequence means one gene, and that every functional gene is only functional if represented by that sequence. That was one of the assumptions. And also he made some back of the envelope calculations, and he concluded that you need, so the space that you have to evaluate to find this unique sequence that is functional is about 10 to the power of 500. So again, calculating, he said, well, if I have a constellation of universes as large as our universe, and since the universe started, I make so I generate one random sequence per cubic angstrom, I don't have time enough to generate the simplest, the tiniest functional molecule. So John Maynard Smith, then coming back to this point, so took this analogy to clarify that, of course, meaning is not randomly distributed, that there are correlations, and that half meaning, so like half an I, is much better than no I at all, right? So you can have partial function, and then you enter the game of doublets, if you want, and then you can move on these networks and optimize function, right? So the situation was not that difficult. So nowadays, we know indeed that genotype networks are vast in the space of sequences. So if you take a protein, for instance, and look at the many different sequences that give rise to this protein, you have a very large ensemble, typically, of different sequences that you can mutate, that allow you to move through those spaces, and that typically percolate the space of sequences. So you can go from one point to another point, and you don't lose meaning, you don't lose function in the way of when you go through this pathway. And so you can explore innovation, you can explore all the phenotypes in this way, and you can get optimized, right? So so far for the say anecdotal introduction, and now let me tell you how we deal with this problem of evolution on genotype networks. So you know that the map from genotype to phenotype is exceedingly complex. You have a set of DNA, a set of chromosomes, and you eventually go to an organism. So there are many layers of expression from one, since you start with your DNA sequence, say, until you go to the fully developed organism. So we cannot, at this moment, study this problem in full. And what we do is we simplify the problem and go back to this idea of mapping sequences into proxies for phenotypes, which might be, for instance, secondary structures for RNA, or 3D structures for proteins. You start with the sequence, you study variations of this sequence, and if they map onto the same phenotype, then you say, well, they belong to the same network, so let's study the properties of these networks. The first thing that we note is that these networks, as Maynard Smith qualitatively suggested, are highly redundant. You start with a sequence, but you have lots of, for instance, synonymous mutations that lead to the same structure. Further, you can have mutations that are not relevant at the level of structures, just change it slightly so the function is maintained. And even at the level of organisms, you may have even gene knockout. So you can eliminate full genes, and there is an intrinsic redundancy in functionality at the organismal level, where other genes, other proteins can partially perform the function of the gene that has been eliminated. So what we know is that the larger the number of levels, the higher the redundancy that you get in your system. So not only at the very basic level of sequence, but also at higher levels, right? And this is a very powerful principle for evolution that gives you robustness and allows you to evolve without losing functionality. Right. What's the architecture of these networks? This is a problem that interested us since 10, 15 years, I don't know. And so the difficulty here is reconstructing networks and knowing if you are close to getting to grasping the actual features of these networks when we are so strongly limited by the numbers. So one of the examples that we have used most often is that of RNA. So RNA has a secondary, it's a sequence of 4 nucleotides, And we can map this onto a simple phenotype, say, which is the folded secondary structure. We can do this through thermodynamical principles, and we pair nucleotides. They are paired and unpaired nucleotides, and we commonly use this representation where parentheses upstream indicate those nucleotides that will later pair with those other parentheses when you go towards the end, for instance, here or at any other position. And points represent unpaired nucleotides. So this is one of the representation. And this we can do systematically. There are algorithms to fold RNA sequences onto RNA secondary structures. So, and here we can now quantitatively estimate the levels of redundancy that we have when going to a secondary structure. For instance, I'm giving here an example with 5 different sequences. They are as different as 2 randomly chosen sequences can be. So the similarity is no higher than about the 25%, right? You will get if you choose them randomly, but all of them fold into the same secondary structure. Okay, how many of these sequences do I have to fold into the same phenotype? Right. Okay, there is a mathematical calculation that you can do that was performed actually long ago in the 70s by Stein and Waterman. And this is an upper bound to the number of different structures in the space of sequences of a given length n, right? You see that the number of different structures grows exponentially with n, and at the same time, the number of different sequences also grows exponentially fast, right? You have 4 to the n. So on the average, if you take this example that I was giving here, for a length of 35 nucleotides, you have about 10 to the 10 different structures, but on the average, you have 10 to the 11 different sequences that fold into each secondary structure. So these networks are absolutely huge. So even for these very short sequences, you have as many sequences for each secondary structure on average as neurons in your brain or stars in the galaxy. And this is a really small sequence, right? If you go to length 1000, the number of different sequences of length n, sorry for the confusion, this is the number of sequences, is about 10 to the 600. So these spaces of sequences are impossible to be explored, never, ever, regardless the computer that you have. So evolution, of course, has not been able to explore these sizes. You see, 1000 is a small gene, right? And we will be also absolutely unable to explore this computationally, never in the future, right? So just keep in mind that the number of particles in the universe is about 10 to the 70, right? So we are so far from that. Okay, anyway, we do our best. So what we do is we exhaustively explore small spaces, relatively small spaces, and try to see if we can extrapolate to larger spaces. So this is our principle. We cannot do it computationally, but anyway, let's do our best. And then let's see if we can also derive some perhaps analytical understanding of what's going on to extrapolate to longer sequences. So this is an example of the kind of calculations that we can do computationally speaking. This is, we have folded exhaustively the space of sequences, don't laugh, please, of length 12, right? So, but even with this very small length, you get networks as large as 60,000 different sequences folding into the same secondary structure. And here we can measure things. These are pretty large networks, right? So you have also orders of magnitude of differences between small networks, small phenotypes. So phenotype secondary structures that have not that many sequences folding onto the secondary structure, and others that are much larger. And we can calculate here in the same vein that I was showing in a former slide. So the number of neighbors, of neutral neighbors that you have, and you can calculate the distribution. So here in blue, you have the distribution of number of neighbors for relatively small sizes, average sizes in brown, and large sizes in black. So this is not telling us much, because what we see is that the average degree grows, and also the dispersion or of these distributions grows. But we were lucky, because it turns out that we could derive for RNA an analytical result that tells that so the average degree of these networks grows as the logarithm of the size. And that's nice, because once we had that, so this relationship was measured in other models, and also, I will tell you later briefly, in real data. And it turns out that this behaves in this way. So that's nice, because this means that regardless the length of the sequence, we have an argument to claim that this is a universal property of these spaces of genotype networks, that their average degree, the typical number of neighbors that a given sequence has in one of these neutral genotype networks, goes proportional to the logarithm of the size of the number of nodes that the network has. Okay, so this is just the theory, the lines here, red lines and black lines. And inside you see the empirical measurements, computational measurements with an RNA model, right? So they nicely fall within the limits of the theory that we derived. Another property of these networks of this case that I'm discussing is that they are assortative, and this means they are correlated in the same way that the meaning of words is correlated and not randomly distributed. You have that sequences that fold into a given secondary structure, if they have many neighbors around, that is to say, they accept many mutations and do not lose the secondary structure that they represent, also have neighbors that have many neighbors in their turn, right? Sorry. So this is assortativity. So high degree is correlated to high degree, low degree is correlated to low degree. This means that for those networks, we have a sort of periphery of low connectivity and an internal core with highly connected nodes, right? This meaning that neutrality in a certain region of my network is higher. So I can admit more neutral mutations if I am sitting in this part of the network than if I am sitting here. Another nice property that we could calculate after many efforts is that the distribution of phenotype sizes is a log normal. This has to do with the distributions that I was talking about. So you have, for the same length, you have so phenotypes which are, say, small, and phenotypes that are 2 orders of magnitude larger. So this can be extended, and this is so points represent data for RNA of length 20. This is an exhaustive fold of all the space. And this is a probability that you find a phenotype with this given number of sequences. Note that here you have the logarithm of the number of sequences. So this is 10 to the 2, this is 10 to the 6, this is 10 to the 9 sequences. So this is a very skewed distribution, where most of the sequences fall into these large phenotypes that have most of sequence space is represented here. And this is, so these are actually very few sequences that fall into small phenotypes. Right, and this is log normal distributions, because this looks like a normal distribution. It is a normal distribution, but in the x-axis, you have the logarithm of the variable instead of the variable, right? So I don't want to go into math, so just to let you know that we can calculate this. And this is another property that we think it applies to any model, because we have tested with all models that we have tested, they have distributions, they retrieve distributions that are compatible with log normal distributions for the size of phenotypes. And in the case of a simplified model for RNA, we could calculate it explicitly, and we got this log normal distribution. So we have an understanding of what's the reason for that. And it's a mixture of mutations allowed in different positions, in different structural positions, and combinatorics of secondary structures in this case, right? So, well, this has 2 elements, and this leads to the log normal. What I'm showing here, just to go into what the evolution says now, is some data that belong to actual non-coding RNA in nature and the distribution that we expect, right? So what are the points? This is sequences of RNA, of coding RNA that they have been taking, some colleagues did this research, they have been taking for the non-coding RNA database. So they are functional RNAs that are functional because of their structure. So there is a method to calculate, to estimate the number of different sequences that fall into each of these non-coding RNAs. And this is what was done here. So this is the distribution of expected sizes of the phenotypes for these real, naturally occurring molecules. What do you see here? Well, the point is that only large phenotypes, and as I was anticipating, are found by evolution. Maybe I have strongly functional or very fit secondary structures here. I do not know, but nature doesn't see it. Why? Because random explorations of sequence spaces can only detect phenotypes that are very abundant. And since this is a logarithm, again, since this is a logarithm, you see that you are bound to explore what is really represented in the space of sequences. So 10 to the 42 is already a very big number, and it's many orders of magnitude above the typical size or the most abundant size of those phenotypes. It's 10 orders of magnitudes more abundant. So evolution is unable to find this, say, rare phenotypes, and is concentrated on functional but also abundant phenotypes. Another property of these spaces is, or of these networks of genotypes, is that they are highly accessible. Why is this so? Because these spaces are highly dimensional. For each sequence of a given length, of length n, you have, so the number of neighbors is 3 times n. So if you have a sequence of length 1,000, you have 3,000 possible neighbors for this sequence. And that's the dimension of the space. So you can go into many, many, many spatial directions. It's absolutely impossible to capture this high dimensionality into a 2D representation. But what this means conceptually is that when you are sitting, for instance, in the green network, right, you may have access to many other different networks, depending on the position you occupy on this green network. So you can jump to the blue phenotype if you're near this area, or you can jump to the red phenotype, or you can jump to the yellow phenotype. And for these extremely large networks, essentially, you have accessible any other phenotype that is also common. So the kind of network we have to imagine for the accessibility of these different phenotypes, it looks more like that than something, say, linear, or that goes from one phenotype to another. Essentially, all large phenotypes are connected to each other. If you are sitting here, you can jump to any other phenotype in your network. Again, this is a result, let me emphasize, of this phenotypic bias, of the fact that some phenotypes that are so large, have that large genotype networks, and also of the fact that the space is highly dimensional. So the 2 key points to have this representation. This is actually a network that we extracted for RNA of length 20, I think. And you see that also small phenotypes are connected to many others, but large phenotypes in particular, which occupy this external ring, are all connected to each other. Right. So you can jump, in principle, if you are sitting in the right position in your neutral network, to any other phenotype, at least so if you are an RNA sequence or an RNA population. All right. So when you go to given environments, these phenotypes have a value that is called fitness. You have to fix your environment, and then every phenotype is more or less fit in a given environment. So we could represent, in a sense, the landscape on which evolution occurs as something like that. You have blobs, so these circles, colored circles that indicate fitness. Size indicates number of different sequences that lead to that phenotype. And then we would have, once this is described, so different probabilities of transition. Large phenotypes are highly trapping. This is why evolution sees preferentially these large phenotypes. So because they can be accessed from many different places. And if they have a relatively high fitness, you are trapped here. And maybe it might be difficult to find phenotypes with higher fitness, but that are smaller in size. That's to say, represented by a lower number of sequences, right? So this is not a symmetrical transition process. If you go to large phenotypes, you have this trapping effect. I'm representing these through arrows, and it would be more difficult to go into phenotypes that are smaller, right? Like so, and if fitness is lower, of course, this probability of transition is really tiny, and evolution mostly occurs on what is large and relatively fit. Right. I'm explaining all these concepts through pictorial representations. I think this is interesting to get the concepts, but I just wanted to tell you that we have also developed a lot of maths for these processes that you can find in the papers if you're interested in. Okay, so some time ago, we also proposed that as a consequence of what I've been telling you, the landscapes on which evolution proceeds should be better represented as adaptive multiscapes, where each layer would represent a different environment. In a given environment, you can define fitness. If you change the environment, fitness is different. The fitness value of your sequences is different, of your molecules. And then once you are on those landscapes, you move from phenotype to phenotype with a very high connectivity between phenotypes and with a sort of arrows, so favoring certain transitions in front of others, right? So not all phenotypes are equally likely, and not all mutations lead to different phenotypes. So the probability of changing phenotype, for instance, depends on the size of the phenotype where you are sitting currently. Okay, let me tell you now a little bit more about the dynamics on these networks. So the picture is the one that I have shown you before. Within each phenotype, we have a neutral network. And as long as the phenotype is good enough, so say fit enough, and the environment is fixed, I have evolution on these neutral networks. So this is called neutral drift. And this is the process by which neutral mutations are fixed. And this is also the process by which, so for instance, quasi-species, viral quasi-species, or heterogeneous populations expand their diversity at no cost for their fitness, right? So they evolve on these neutral networks and they can acquire a larger number of variants. This is also what influenza does. I mean, there are lots of examples of how this proceeds in real systems. Right, but as I was saying, this is in a situation where your phenotype is relatively fit and you don't change the environment. Of course, you can have adaptation through mutations. You might have mutations that occasionally lead you to another network and allow you to jump from one phenotype to another. This usually entails some population bottlenecks. So the diversity in the population shrinks, temporarily shrinks, and you jump to a new phenotype, and then you expand again in the neutral network of the new phenotype. So you have these sort of contractions and expansions every time you find adaptive mutations. And expansions are related to the number of neutral mutations that you accumulate, right? So, but when you have environmental changes, then you jump to another layer of this landscape. In the other layer, you may have some sequences, some molecules that may express a different phenotype. This is called phenotypic plasticity. So not necessarily the same phenotype is expressed. For instance, in RNA, if this would be two environments represented by two different temperatures. So at one temperature, you get one fold, and then a different temperature, you get a different fold for RNA, right? So you map into a different phenotype. And the fitness value in the new environment would be also different. So the process would start again. But something that is very powerful in adaptation is that without the need of incorporating new mutations, you change your environment. And some of the sequences that were already sitting somewhere in your neutral network might be, say, sufficiently adapted so as to start the process of adaptation in the new environment. And again, you would have this change of phenotype, so neutral networks, accumulation of mutations, advantageous mutations, jump on phenotypes, and so on, right? Okay, so in my view, this is a much more natural way of visualizing the adaptive process. It may not be as simple as the fitness landscapes we are all familiar with, with mountains and valleys, but this is much more realistic, because this is telling you that you can jump to many different phenotypes, not just to the phenotypes that go uphill, but to many others. And this also takes into account changes in the environment that are very different to capture in the other kind of landscapes, in right landscapes. Right. Okay, so what happens when we are evolving on a neutral network, just inside the same phenotype, neutral network, neutral mutations? So the population of sequences then tends to occupy areas of maximal connectivity. I was telling you about these correlations. You have a periphery with low connectivity and the core, and there is a gradation, of course, of different connectivities in between. But just to simplify, then your population and now the size of these nodes represents the number of sequences that I have. Hope it doesn't, it's not confusing. So the population tends to sit preferentially on the regions where the connectivity is higher. Why? Because this is a way of increasing fitness, actually. So here, mutations still maintain the population in the same phenotype, while here mutations send a larger number of sequences to non-viable or to less fit phenotypes. So the population tends to sit here. This means that robustness to mutations increases along neutral evolution, and this is a relatively old result that we knew about. But since my networks are assortative, and that was a new observation, evolution of neutral networks actually what does is accelerating the ticking rate of the molecular clock. Why? Because when I am sitting here, I can accumulate few new mutations without changing a phenotype. But if I'm sitting here, I have more possibilities for accumulating new mutations. So my molecular clock here ticks slowly. And as I move inwards towards these more connected regions, the ticking rate accelerates, right? Goes faster. This means that there is a correlation should be a correlation between the length of phylogenetic branches if you have not changed your phenotype and the ticking rate. So the rate at which mutations accumulate. So the molecular clock is a sort of relativistic clock that depends on time and is not Markovian. Well, this is something that has to be taken into consideration, although for our calculations, it seems that the effect is not that big, right? So as a first approximation, a regular ticking clock might work. Another property of these networks, and this happens, as I will tell you, at the different levels, is that they have punctuated dynamics. So adaptation proceeds in jumps. At the level of the neutral network within a phenotype, this is the same case that we were discussing previously. So this happens because you have a community structure in these networks. So you have a hierarchical construction of these networks. At least for RNA, what we have is that regions that are most densely connected, that have more neutral networks within that region than with other regions of the network. So you have this hierarchical construction, so connected regions, then a second level of more connected regions, and so on. And the result of that is that when you have populations evolving on a neutral network, what you see is that there are discrete adaptive jumps, even increasing only, so average fitness based on neutral mutations, right? So that there is no change of phenotype here. I cannot say there is no adaptation because these regions, these more connected regions, are more robust to mutations and they are favored by selection, right? So this is punctuated dynamics. So similar to the punctuated dynamics that we all have heard of regarding the macroevolution and the fossil record, but this happens already at the molecular level, right? And in neutral mutations. Not only in neutral mutations, it also happens when you change phenotype. Why? Because you have this shrinking of diversity and expansions of diversity that I was discussing previously when you change phenotypes. So this is an example where you start with a random population of RNA sequences, and then you want to reach a goal secondary structure. So what you do is you select your, so those sequences that fall into structures that are sufficiently close to the goal structure, right? So in principle, they do not resemble it so much, but some do it better than others. And then you fix these variations, and then you start exploring your network. This is the average population structure distance. And this is the dispersion, a measure of the dispersion of the population in the space of genotypes. So to say, the diversity of your population. So you, for a while, maintain your phenotype, well, but something is happening at the microscopic level because your population is more diverse. And then you have these jumps when you encounter a new phenotype. You have this shrink of diversity, and then followed by another expansion until you localize in this space of networks another network with a fitter phenotype, and so on and so forth until you reach your goal, right? So these jumps in adaptive evolution at the molecular level were proposed also long ago with RNA, so with, say, toy models that, however, are not trivial. But it was demonstrated later in the 2006. You have this extremely nice paper in Science by the group of Mercedes Pascual, where they were measuring this behavior of expanding in a neutral network, jumping to a new neutral network. And this was correlated to antigenic shifts in influenza A. So this is absolutely the way in which adaptation season to season of influenza proceeds. You have these expansions, reduction in diversity, new expansions, and localization of new phenotypes that can escape the effect of immunity of the immune system, right? So this is a general process in microscopic evolution, right? And also you can have these sudden jumps, this punctuation, if you have environmental changes. We proposed that this behavior, that this should happen some time ago with, again, with a proof of concept model, where actually what we just took networks, and what we did was for each node in the network, we associated two different features that were competing. So in one of the networks, in one of the regions, we were favoring high robustness, so higher connectivity, as neutral networks do. And in another region, we were favoring low energy of the fold. For instance, this would be preferred if you are folding at a very high temperature and you have to maintain your structure. You prefer folds that have lower energy because they are less disturbed by increases in temperature. So there is a way in which you can balance one selective pressure versus the other. And then you can calculate, so this is the parameter here, beta over mu, doesn't matter what it means. This just says that I'm giving more relevance to high robustness here and more relevance to low energy at the other side of the plot, right? And in between, what we saw, it was that there were sudden transitions between the two states. So essentially the population maintains one of the states until it goes to a point where both selective pressures exactly balance. And then very rapidly, it goes to another situation where the other selective pressure is dominant, right? So this is not a gradual evolution. It's not that through time, the population slowly changes from one position to the other. So you have environmental changes, and this you can relate to climatic change as well. It's a bit far-fetched, but the idea is you have sometimes things that change very slowly as environmental temperature, and all of a sudden you see violent transitions in your system, right? And this is what happens here also at the genomic level. And this is also the time it takes for the population to equilibrate to the new state. And this is a typical phase transition of physics, where the time to equilibrium, well, we don't know if it diverges. We think it does, but what we see here is a tremendous increase in the time it takes to settle into mutation selection equilibrium, which is what we are measuring here eventually. right? So we have these three different situations, and all of them lead to punctuated changes at the molecular level, right? This is another representation of this last situation where we have so a different visualization actually of the network of genotypes. This is well, a certain kind of landscape of an NK Kaufman landscape if you're familiar with, otherwise doesn't matter. These are the different sequences that you have in your system, and this is the abundance of the number of sequences that actually sit on each of these particular sequences. So you have this here, this sudden transition when smooth changes in the landscape are performed. So this is a bit more realistic than the previous case. It was much more academic. But if you look at either this representation of the nodes and the fitness of the nodes, when you change this parameter that represents tau, represents the changes in the environment, you see that not major changes are observed, right? More or less, you keep more or less constant noisy fitness for each of the nodes. But if you look at the abundances of the population, you see that it is preferentially occupying these nodes here, where you see high population numbers. And then all of a sudden, it jumps to another situation where, so the preferred situation is somewhere else. So you see smooth changes and sudden responses. This is a highly nonlinear system that is also interesting because it's telling us this is one more example of what happens in many other complex systems where small changes do not lead to small causes, but big causes, right? Okay, so I'd like to finish. Let me see how much. Oh, I've been talking for a long time by now. So very briefly, I want to tell you about data because everything that we were doing in the last 10 years mostly was computational. But now we thought, okay, we need to know where populations actually sit. What we have done is in collaboration with one of our colleagues that has an experimental lab, we have investigated the case of the cubic phage and its adaptation to different environments meaning different temperatures. So this is a procedure that can be carried out of the lab. You have a population and you undertake different passages until you get an evolved population. And for instance, you keep constant the temperature at which you do that. And what we have done has been to deep sequence the population at different points of this evolution. The cubic phage is, this is a picture of the cubic phage, a representation, not a picture, of the capsid, and it has just 4 proteins. It's a relatively small phage virus. Okay, so we have different experiments of adaptation of the phage at different temperatures. The different lines indicate how many passages and at which temperature these passages have been performed. And what we have done, as I was saying, has been to deep sequence the populations of the phage, certain regions at different points of this evolution. So we have chosen 3 different regions in the genome with lengths that are bound by the experimental, but the technical ability of sequencing unique sequences, right? So they go from 270 to 330 nucleotides in the different regions. And this is the size of the networks that we have now, that we are analyzing now. So hundreds of thousands of different nodes, and this means different sequences that the phage has in its populations, and about twice the number of edges, so the connections between these nodes. Right? These are huge networks, and what we have done has been to reconstruct the network. So take the sequence, and if you have a second sequence at the distance 1 mutation, you have a link between these 2 genotypes, and so on and so forth. This is how you reconstruct the network. And this is, for example, a picture of how the population moves in the real sequence space. So we were looking previously at computational results, but these are real sequences. So the links you see below in black are the reconstructed network, and the points are the top 100 most represented sequences at each time. X-axis is time, number of passages, and y-axis is the temperature at which the system has evolved. And you see here nicely, for instance, if you look at the last passage, that if you change temperature, you go from a preference for a certain region of genotype spaces to a selection of a different region of your genotype spaces. So your population has moved. Whether this entails phase transitions, we do not know yet because we have a few points. So our next plan is to sequence actually, so not look for the transitions under environmental changes, but look for the transitions in adaptive changes, which correspond to this line here, right? And see if my population at some point jumps suddenly or not parsimoniously, say, from one situation to another. Right, these are experiments that we are currently undertaking. We still need to sequence more, and what we have done up to now has been to develop the algorithms to reconstruct these huge networks, which is not trivial, and to project the different experiments that we have. Right. Okay, so let me go to the conclusions and see if you have any questions. So I'd like to visualize the space of genotypes as a multi-layered network of networks that as I have shown you, right? So we have some mathematical results regarding how evolution should go in those networks, but there is still much work to be done. There are three situations that link to punctuated dynamics, which was when you increase robustness, when you adapt, or when you respond to environmental changes. There is something that I didn't have time, I didn't want to go into, but this is this punctuated behavior can be described mathematically formally as a competition between networks, which puts this behavior into the realm of complex networks and related phenomena. Okay, so and then we are now at the time of measuring these properties of genotype networks with deep sequencing data and see how they differ, if they do, from the theory we've been developing. So I'm certain I know already that we will have surprises and we will learn much more about how molecular evolution proceeds, right? So if I have to tell you a take-home message, this is my take-home message. This is not a good representation of sequence evolution, right? So this is, if you have this metaphor in mind, it will be very difficult for you to interpret correctly empirical results of adaptation in viruses, in viroids, in RNA sequences, and so on and so forth. So my acknowledgements to my colleagues. I've been working on the topic for many years with many different people. If you are very strongly interested in the topic, there is this review which is to appear. But let me insist, you should be very interested because this is a 100 pages review that, of course, will tell you the state of the art and the perspective of the field, right? If you are not that strongly interested but find it appealing, there is also this publication in Investigación y Ciencia for the ones of you who speak Spanish that, well, we published it last April in the midst of our confinement. And it's also a nice and illustrated account of how evolution proceeds on these genotype networks. Okay, thank you. I'll be happy to answer questions if there is any. So,
thank you very much, Susanna. That was a really fascinating talk, and I liked your take-home message. I'll keep that in mind for the future.
That's great. That's great.
Okay, there are no questions in the chat yet, but please do write your questions there. We are all keeping an eye on the YouTube chat. And while we wait for people to write down a question, so I have one question about the methods. I really like the visual representation for the results you had, because I think for a talk that's just brilliant if you can actually communicate your mathematics without having the mathematics on the slide. But just because of my personal background, what sort of methods are you using? Is that mostly graph theoretic, or is that probabilistic?
No, it's mostly graph theoretic. I mean, for instance, to represent the evolution of populations of networks, what you need is the connectivity matrix of your graph, right? And maybe I have some. Yeah. So you have your a matrix that tells you about. So this has a C is the connectivity matrix, for instance. E is in case you have a fitness landscape. If you are on a neutral network, this is an identity matrix, right? And then you have a mutation between neighbors, right? You have mutations, you have a replication. This is replication, right? And you have if you include your fitness landscape, you have fitness, right? So and then once you have this matrix, you know, for instance, that mutation selection equilibrium is represented by the eigenvector that corresponds to the largest eigenvalue of this matrix, right?
Okay.
So from then on, you can calculate, for instance, this is a representation where you have a decomposition in terms of eigenvalues and eigenvectors. This is a projection of the initial conditions that also appears in the solution. And then you can say, so decompose your matrix and say something about, for instance, the dynamics. Something that we can say is that the time to equilibrium, for instance, goes as the inverse of the mutation rate. This is the kind of derivations you can make from that. You can calculate the spectrum of eigenvalues of your matrix.
Okay.
And there are some also interesting results regarding how fitness and robustness compete when both are present, because the eigenvector, of course, takes both into account. Both are weighted, right? You have fitness and topology, and you cannot disconnect them. But you can say something about this competition between networks, right? So here you have, for instance, an example of how this competition would be represented. You have a matrix of connector nodes, and then you can do perturbation theory to go from one eigenvector to the other eigenvector when you change some of the features. And while you have transitions that depend on the distance between the eigenvalues, and yeah, this is the kind of a scheme that we have, right?
Oh, really interesting. I definitely have a look at the article you mentioned
there.
Okay.
There are other things, for instance, to calculate this log normal distribution. This is combinatorics. But this is advanced combinatorics, actually. So this is done with methods that resemble, in a sense, Feynman diagrams. So because you have an inductive way of calculating when you add new, so you enlarge your sequence, for instance, right? So this is also published somewhere. So if you go to my webpage, any of you that is interested, or just drop me an email and I'll tell you. Right.
Okay, brilliant. Yeah. Okay, we have questions from the audience.
Good.
So we can move on to that. So there is one question from Francesco Marchetti. Thanks for the very interesting talk. Is your work related at all with AlphaFold?
No, no. I mean, maybe AlphaFold will solve some of the problems related to protein folding, but not to our case, because we are more interested in the exhaustive exploration of spaces, right? AlphaFold is directed towards solving a long-standing problem, which is how proteins fold. So until now, for instance, for RNA, these thermodynamical principles work relatively well to give us some intuition about what are the secondary structures, right? Just taking the sequence, but there was up to AlphaFold, there was no good method of estimating folds of proteins from sequences. And so, of course, this is an advantage in the field, an advance, but I don't think I will take advantage of that improvement. Yeah.
Okay. There is another question from Gabriela Ochoa. Can you have phenotype networks of structures that are more complex than RNA secondary structures?
Yes.
And if so, how to uniquely represent the phenotypes so they are nodes in the network?
Yeah. Phenotype is defined in the way you prefer. So what we know, because we have not only investigated RNA, we have also investigated systems that have multiple layers, and there are also results for empirical results for that map short sequences, for example, binding tRNA sequences that bind to certain parts. And then you measure function. You are not measuring a secondary structure. You have different definitions, right? And the good point is that most of the properties that I've been describing do not seem to depend on the definition of phenotype. So there is a sort of universality in the topological behavior of genotype to phenotype, free phenotype definition maps. Right, and so we believe that what underlies is a stronger principle, which is that of navigability. So we have to go back to Maynard Smith and say anything that is found in nature has to be navigable, because otherwise natural selection cannot act on it and it will be lost. So this is a sort of first principle. And if you require this navigability, then you fall into a universe of similar minimal topological properties that are shared by all models and by real systems we understand at this moment. Right.
Okay, great. There is another question from Bill Langdon. Are there quick ways to estimate first 2 or 3 eigenvalues?
Well, this is a mathematical problem. So for our matrices, we didn't have much troubles actually with using available software. But this is not a question that I can answer. I guess that mathematicians are interested in doing that for huge networks probably, or people involved in numerical computation, but that's out of my knowledge. So what I can say is that we were able of calculating at every time the eigenvalues.
Okay. So I think there is one final question in the chat, so I just ask it. It's from Nuno, who you've met earlier. What do you think of the feasibility of using the data of the networks to anticipate or predict the regions where the networks will be? Is it, and will it be possible?
Yeah, that's a hot problem now in evolution, which is the predictability of evolution, right? I guess that the question goes in this direction. Yeah, that's an amazing question. And I think that now more or less the consensus is that if you are placed locally in your space of sequences, so you have your population, it's more or less fit to the situation where it's evolving and replicating, then there are not that many pathways that the population can follow. So there are preferred mutations and preferred pathways. These people have been observing it in cancer and with these driver mutations and in other systems. So there is a strong sense of phylogeny in evolution. So you are strongly conditioned by the position where you are. The question is whether this will happen at very long evolutionary times. And that's not so clear to me. So if you allow enough time, then maybe these pathways diverge, and then you can finish in very different regions of your space of sequences, right? This we have seen for smaller organisms like viruses and viroids probably. So the homology in sequence is lost after a certain time. But locally, yes, there is a sort of convergence of pathways that allow you to at least do some probabilistic prediction of what is more likely.
Okay. Okay, really, really interesting. Okay, I think that's it for questions. So just there is a lot of virtual applause in the chat.
Oh, thank you very much.
And positive feedback about the visual material and the gorgeous presentation, to just quote one comment. So
I'll stop sharing my screen because I had here the
so you can probably see that yourself.
Yeah, I can see now. Yeah, right.
Okay, yeah, thank you very much again. I think that was really, really interesting and a nice new perspective.
Okay, hope you enjoy.
On that sort of topic. I really enjoyed it, and I think so did the audience. So
okay, thank you very much.
I have a single applause. Okay.
Hope to meet you in person at some point.
Yeah, of course, yes. That would be really nice. Next year, maybe.
Okay, maybe. So you all take care.
You too. So I was asked to just relay one more announcement, because we will have the closing session now, and obviously if you want to join us, you're invited to that as well. It will take place in room D on Zoom, and the instructions were sent by email, so everybody registered for the conference should have the instructions somewhere. So I hope to see you all there in a couple of minutes. Okay, and thanks